The next speaker will be Noga Zilkha from Weizmann Institute of Science, and she will talk about we are what we eat, which is picky, I must say. Anyway, genetic basis of the effect of nutrition on autism-related behavior in mice.
Okay, so thank you, Shlomo. Thank you, everyone, for inviting me and for listening. My name is Noga, and I'm a research associate at the lab of Tali Kimchi at the Weizmann Institute. And I'll be presenting two of our projects regarding the effects of nutrition on autism-related behavior in mice. It's been commonly known that obesity and high-fat diet causes major disruption in metabolism and many physiological disruptions. And it's been also been commonly known that these effects also cause major behavioral impairments, including impairments that are related to autism. And in our projects, we first try to assess whether the effects of high-fat diet, meaning a diet that is consistent with 60% fat, affect autism-related phenotype. For this purpose, we use the BTBR mice, which are a known mouse model for autism. We took males from this mouse line and kept them from their own weaning onwards on either high-fat diet or control diet. And when they reached adulthood, we gave them a series of behavioral assays, including the resident intruder assay, which tests for social interaction, olfactory preference for social odors, social preference and memory, and the wet T-maze, which tests for cognitive rigidity. And I won't show you all the behavioral data because it's already been published and due to the lack of time, but I will show you that when we comprised an autism composite score from the standardized behavioral assay, we saw that the high-fat diet mice received significantly higher autism score compared to the controls. And we also saw that this score was correlated to their weights, meaning that the higher their weight was, the more severe their autism-related phenotype was. So this was the first project, and in the next project, we tried to assess the effects of maternal high-fat diet on autism-related phenotype, and for this purpose, we used this time C57 mice, which are considered neurotypical control for many autism mouse models. And this time we took females of the C57, and from their weaning onwards, we kept them on high-fat diet. We mated them, let them deliver offspring, and when the offspring reached their own weaning, we moved them back to standard laboratory chow. When the offspring reached adulthood, we conducted the same series of behavioral assays. So first, I'll show you the result of the social three-chamber assay, which tests for social preference. The mice are in a three-chamber apparatus. On one side, they have an empty cage. On the other side, they have a similar cage containing a mouse. And a typical mouse will prefer the social stimulus over the empty cage. And this is indeed what we saw in our control mice, which significantly preferred the social stimulus. However, the high-fat diet offspring did not display a significant preference for the stimulus. We next conducted the resident intruder assay, where we first saw that the percentage of animals displaying aggression was significantly higher for the high-fat diet offspring compared to the controls. And when we further quantified their behavior, we saw that the high-fat diet's offspring spent significantly more time in aggressive attacks and significantly less time performing olfactory investigation or sniffing. We performed another assay in the three-chamber apparatus, this time with an olfactory social stimulus, where on one side the mice receive clean bedding, on the other side they receive soiled bedding from either males or females on alternating days, and the typical mouse will prefer the social stimulus, and also males will prefer the female stimulus. when we quantified the results, we saw no significant effect in the preference for the male bedding, but we did see a significant reduction in the preference of males for female soiled bedding compared to the controls. We next wanted to try to find out the molecular basis or the neural basis for these behavioral effects. We took samples of the olfactory bulbs for two major reasons. First, it's a relatively large brain region and also relatively homogeneous. And also because we saw many of our effects in olfactory investigation behavior. So we took samples from both groups for a microarray and received a list of genes that had altered expression between the two groups. We then further validated the results using real-time PCR. These are the genes that were validated, and we saw significant increase in two of the genes from the microarray, the ERDR1 and the CHRD1. We also tested a few more genes that are related to social behavior and neurotransmission, the oxytocin receptor, the dopamine D2 receptor, and the tyrosine hydroxylase, or TH, which participates in the synthesis of dopamine. We further looked at the hypothalamus, this time only with the real-time PCR, and we saw again an increase in the expression of ERDR1, and interestingly, a reduction in the expression of TH. This is where we're trying to go further to see whether the reduction in TH is related to the increase of aggression, which relates to a previous study from our lab. To summarize, a high-fat diet worsens autism-related phenotype in BTBR mice. Maternal high-fat diet reduces pro-social behaviors and increases aggression in male offspring. And maternal high-fat diet also reduces hypothalamic TH expression, which is where we're going next. And I would like to thank all of you for listening, the organizing committee, and of course, everyone in the Kimchi Lab and the funding agency. Thank you.
Thank you, Noga, for the appetizing lecture.